Okay, let's pick up where we left off. AP review for the second semester, and this is part 3. So we kind of left off talking about some DNA technology stuff. We're going to continue with that, and then we're going to do some evolution stuff, and then fourth video, do some comparative anatomy pieces. So we left off talking about how you could do gel electrophoresis to separate DNA based on its size and how you can radioactively tag. One other thing that you need to understand is what we call PCR, polymerase chain reaction. So basically what this is all about is DNA or RNA amplification, where we are going to do this in vitro. So we're going to do this in a test tube as opposed to allowing a cell to do it. And in order to do that, what we need is our original piece of DNA. So you can take a small piece of DNA and you can amplify it. So by running a heating and cooling cycle, use a thermocycler to do this, hence its name, thermocycler. You're going to run this amplification. It's basically like DNA replication, but we're going to do it in an artificial kind of way without cells. But we're going to still use enzymes. So you're going to need DNA polymerase to do this. But the problem with DNA polymerase is it's temperamental with its temperature. So you need one that's heat resistant. So we use one that's called TAC, which stands for Thermoaquaticus, or Thermophile aquaticus. So they come from hot springs. They're bacteria that live in hot springs. So their DNA polymerase is less temperamental to heat. So they will be able to work. So the reason that we heat it is to separate the DNA, because heat will break hydrogen bonds. We know that back from the water unit. And it separates the DNA. So now you have your exposed piece of DNA, and you can get in to do the complement. Remember, semi-conservative model. So you're going to use DNA polymerase, throw that in there. You're going to add your nucleotides. So you're going to need nucleotides of A, T, G, and C. And then you're also going to need some ligase so that you can glue pieces together. And basically all you do is put all those pieces together, run it at a heat, cool, very coordinated pattern, and eventually it's an exponential growth. So you might have a small amount and then it just keeps going. So you can end up having, after an 8 to 10 hour cycling, you can have millions and millions of copies of the exact same sequence. So why is that really important? Well, if you take a mouth swab or a blood sample or semen sample or any other kind of sample from an organism, even if the organism is maybe recently deceased, you can take that DNA, small portion, and you can run that amplification or run a battery of different tests. So one of them is going to be what we call RIFLIP analysis. So RFLPS. RIFLIPs are restriction fragment length polymorphisms. Basically what we're saying is a majority of organisms are the same within the species down to 99.99% of the exact same DNA, only differing on 0.01%. So that 0.01% we're going to optimize with restriction enzymes. So if you see here, we have allele number 1 and allele number 2. They're slightly different in this section right here. I don't know if you can see it right here. And because of that, the restriction enzyme does not identify allele 2 for a cut at that location. So what you get is allele 1 will cut in 2 places, yielding 3 pieces. So you get this really small piece, you get this large piece, and you get this medium-sized piece. And that's why you see small, medium, large. So they're not orienting based on their location within the chromosome, they're orienting based on their size. So this is a gel electrophoresis. It's kind of hard to see, but this is the positive end. This is the negative end. Negative because DNA is negative, it pulls towards the positive. So the small pieces move the farthest, the fastest. So you got this small piece way down here really quick compared to the large piece. So allele number 1 shows 3 bands, whereas allele number 2 does not get cut on the first sequence that the allele number 1 did. And so it only gets 1 cut. So it gets a small piece. So its small piece is going to match the small piece of allele number 1. But then it gets this really big piece. And hence why you get this Z right up here with that really big piece. So we can identify the differences between species or the similarities. We could identify if a child has the same restriction sites as parents. They should share about half and half from both parents. We could identify unknown samples to known samples, so individuals that maybe left something at the scene of a crime. And then you can test for diseases, right? So those are the four big ones that we use this process for. But once again, you got to be able to amplify your DNA before you do it. Changing direction slightly. It's really important that you understand that cells need to be able to talk to each other. So this comes back to kind of the operon component, but we need to understand that cell number one, way up in the head, can talk to cell number two, way down in the toe, via things like hormones. Right? So hormones can be released, or proteins from one location travel through a blood system, be at a cell's surface carbohydrate and be received, cause this transduction pathway of a bunch of different reactions, and then eventually get a cellular response out of this situation. So it's a very simple process: receive, transduce, and then respond. But this is how all cells communicate to each other, both within a multicellular organism, but also between different organisms. So we talked about how that sensum quorum thing works, with bacteria being able to talk to the same species and figure out if they're at a critical mass to mount an attack, or whether or not there's other individuals in the area that might pose a threat or cause issues. So even bacteria can talk to each other. And that's really big for what we call the microbiome, right? All the bacteria that are inside your digestive tract and on the surface of your skin on the outside of your body. So all those cells kind of communicate, and they identify what's going on in the environment. Your body responds accordingly. Not only is it a hormone situation, but it's an immune system thing. So the ability to respond to cells that are yours versus cells that are not yours is all a cell communication piece. I don't know if you remember us talking about how, bacteria, they have antigens on their surface, and viruses too, on the surface of their cells, and then those pieces will bind with some of your cells, and your body can identify whether or not that's a foreign invader or not. And foreign invaders we destroy. The problem is sometimes our body thinks our own cells are foreign invaders, and they try to destroy our own cells. We call that an autoimmune disease. So cell communication is really, really important. It's a crux of biology, and one of the pieces that you need to understand, especially for AP and the future. We then went into some really cool, interesting new pieces in the second semester. That was epigenetics, RNAI, and CRISPR. We're going to kind of pack them all together here. So epi means above, and then genetics means the genes. So above the genes, what we're saying is that things that you consume or you're exposed to can change which sections of your DNA are exposed or closed down for transcription and translation. So we talked about methylation and acetylation. So methylation, if you have a piece of DNA and it should be transcribed, you can get methyl groups that will attach here and prohibit that gene from actually getting transcribed and translate. And then acetylation, if you remember, DNA wraps around these proteins called histones. When it's packed up like this, the RNA polymerase can't get in there to transcribe. So they have to be loosened out. So you can kind of see in this picture, it's loosened out. So the gene would be exposed here. So different types of epigenetic factors like acetyl groups, can loosen that so that the gene can be exposed to get transcribed and translated. And this is probably why we see, our DNA pretty much doesn't change other than through mutations throughout our entire life, but yet we change throughout our life. So genes that might be active in an embryo or in a young child are not the same genes that are active when we become young adults and older adults. Okay, so that's all based on some of this epigenetic components. RNAI is this concept that we can destroy mRNA before it actually gets translated. So the concept here is, here you have this really long piece of DNA. This DNA piece has genes on it. It turns into mRNA, turns into eventually protein. We have ways that we can cause a destruction of the mRNA before it actually gets there. So there's two main big workers. One is dicer. So if you take a piece of double-stranded RNA that looks like the gene that you want to turn off and you inject it into a cell, dicer will look at it like it's a virus's piece of DNA or RNA, and it chops it. And then you get this small interfering RNA, this siRNA, and that siRNA is then gobbled up by RISC, and RISC uses it as a template to figure out what mRNA it needs to destroy. So then it finds any mRNA that looks like that template, and it cuts up that template. This is why it's called RNAi, RNA interference. So it's destroying RNA before it could actually turn into a protein. So we've had some new advances with this for Huntington's disease. We've had some glaucoma hope with some of this, but they're a different way of medicine than just giving a different chemical compound. We're actually injecting pieces of DNA to try to enact an immune response within your cells that we originally found in bacteria. And then CRISPR, by far the newest one, is this repeat sequence. It's very similar to this scenario where you have this enzyme called Cas9, which is extracted from bacteria. And then we produce this guide mRNA sequence that looks a heck of a lot like the gene that we want to cut out. The cool part is we can cut out DNA. Instead of working with RNA and destroying RNA, we can actually cut out pieces of DNA very specifically. So anywhere where this piece of DNA gene is found, we can chop it out. So if you have a gene that you know is mutated for, say, something like diabetes or any cystic fibrosis, you can put in a guide piece of RNA that looks like that gene for cystic fibrosis, inject it with the Cas9, because the Cas9 is going to do the work. It will use that guide RNA, find the gene that looks like cystic fibrosis, and it will chop out that gene. And so then you have it totally exposed. If you inject this plus a correct sequence of the gene you want to go back in, you can, in essence, do a Control X, Control V situation where you Control X cut out the segment of your sentence that you want to chop out, and then Control V paste in the segment that you would like to edit in. So this is, once again, CRISPR, and this is a brand new technology that we've seen some really good, significant breakthroughs, especially with sickle cell anemia. We've had a lot of patients recently that have been pretty much, it looks like, cured of sickle cell anemia and not needed to do as many blood transfusions to keep themselves healthy. All right, on to the next. So we're going to start looking at evolution. So one of the things with evolution is you need to know the basics of what we call the tenets of evolution. And so what we understand is traits are inherited. We now know that they're inherited in packets called genes, but they're also, it's a chromosome thing as well, right? But they are inherited. So they are passed from generation to generation. So from offsprings to children, or from parents to offspring. And what we do know is that inherited traits have differences, right? So there's variety within a population. We also know that there's oftentimes way too many offspring made for the space that's there. So if you remember, we have logistic growth curve and we have a carrying capacity. Oftentimes, more children are produced than the environment can handle. So eventually it pulls it back down to that carrying capacity because there's some sort of struggle piece. So there are oftentimes better traits in a population for the given time period. And so what we see is those individuals have a higher fitness. Remember, fitness is the ability to survive and produce viable offspring. And so what we have is, after we have this unequal ability to survive in our differences in our pool, that those have a better fitness end up producing more children. So we get an increase of what we call better or higher fitness traits in the future. So we're not saying that the offspring are necessarily going to be better than the parents, but whatever traits allow those parents to survive and produce children, we're going to see more of the children with those traits, right? And that is what we call difference in survival or difference in reproductive success, right? Or what we call survival of the survival of the fittest. But keep in mind that that does not mean that the fittest only survive, right? We use that as a general terminology that those that have a higher fitness value generally produce more children. It doesn't mean if you have a less fitness value that you don't produce children, you just might have a harder time producing children. So those are the basics. And then we started talking about like different evidence pieces, right? The Earth is old; it's constantly changing, right? So if it's constantly changing, we're going to see what's fit is going to change as the environment changes as well. So what might be good now might not be good in the future. We also see, oops, this got kind of moved here. Another piece of evidence is artificial selection. We talked about this with, for example, dogs, any fruits and vegetables, all selected for. It's hard to even go to the grocery store and buy something that hasn't been artificially selected for by humans. So we're choosing individuals that we want to produce offspring, and continue push the curve. A lot of times, in what we call a directional selection pattern. We'll talk a little bit more about directional selection in a second. And obviously, we have copious amounts of fossils, which are traces of long deceased organisms. I learned this in ologies, that fossils are, you can't really call it a fossil unless it's over 10,000 years old. So otherwise, I guess they just call them artifacts. I don't know. I don't know. That's a dig question. But fossils, old remains of extinct or long past organisms. They don't necessarily always have to be extinct. So we looked at what we call homologies. So homo means same, logos means number. So homologies, we're talking about same numbers, same patterns. So we looked at the four limbs of a whole bunch of different organisms. We have this one bone, two bone, a lot of bones type of situation. Dr. Shubin talked about this when he was talking about Tiktaalik, right? And he talks about it in your inner fish as well. But we have this humerus bone that's connected to the ulna and radius, and then we have the carpals, and then the metacarpals and the phalanges. All these organisms here have the exact same bones, the exact same order, but they are used for different purposes, for different adaptations. So they must share a common ancestor somewhere back in the tree of life that allowed for these genes to keep getting passed, but because of environmental pressures, they're used in different ways, right? And this is also connected to what we call vestigial organs or structures. And so a good one that we talked about was the pelvis and the femur that we find in whales, although it's not even connected to any legs anymore. We talked about the eye sockets of salamanders that live in caves. They don't have eyes anymore. They just have flesh that covers that opening, but they still have the socket in their skull. So all those are considered what we considered vestigial structures, trace structures, and they show homology to organisms that they're related to. Excuse me. I also talked about embryos and how as embryos develop, we see similar types of patterns, which means they have similar what we call Hox genes or body plan genes. And those body plan genes allow for the development of the organism. So a lot of us, when I say us, I mean animals, from birds to turtles to humans, all show very similar embryonic shaping, from little eye spots, the little paddle hands, little pharyngeal slits in the sides of our face, tail that's prominent there. And as the organism develops, different genes turn off, other genes turn on, and you end up getting the organism that you're familiar with, right? We also look at DNA comparisons from, for example, like a human to a chimpanzee or a bonobo chimp, right? All those, or even to a fruit fly, right? You look at how much of the DNA is exactly the same, right? The coding for the same proteins, and that would show homology. And then one of the last things we talked about, and I should talk about analogous structures too, is biogeography. So the displacement of organisms, or I should say, the spread of organisms across a given area. So I think we talked about how in South America, this is my crude picture of South America, and then Africa, right? We see organisms on the west coast of Africa, fossils that are tetrapods, the exact same species that are tetrapods on the east coast of South America. So that's a biogeography. So it's distribution of organisms. They're not just willy-nilly placed. They have to migrate in order to get to those locations. So biogeography is a good reference for us for geological distribution of organisms across the mainlands and even water for that matter. We then should talk analogous structures as well, right? So analogous structures are ones that function in the same way, but they have a farther back pass. So something like a bird's wing and a bee's wing. They both serve for a similar type of niche, similar type of adaptation to be able to fly and move through the environment, but they have very, very distant ancestor to each other. A bird is much more, a penguin is much more related to a sparrow, even though the penguin doesn't use its wings for flight, it uses it for swimming, and the sparrow uses it for flight, right? But they're both birds. Okay, so that'd be an analogous structure. Kind of finishing out some of this stuff with evolution, we talked about Hardy-Weinberg equilibrium and being able to apply mathematics to allele frequency changes. One of the big things we talked about was these five things. These five things are pieces that, if they are all true, we would see no evolution. We would see no allele frequency change. So that was, remember, the five fingers, how it connects to the five fingers, where no net mutations, that was the middle finger. No natural selection, that's the thumb. Large populations, that was the reverse of it was your pinky finger, right? No gene flow, that was your index finger. And then random mating is your ring finger, right? So if we see these five things held true, every single one, we will not see the population evolve. So we even say this because there's no way to see a population have all these five things held true. Well, that's because all populations are evolving. So in a sense, we're just drawing a line in the sand to try to determine how much maybe a population would be evolving based on these main premise. Okay. Patterns of selection, one of the last things. We should do sexual selection as well, and a little allopatric speciation. Patterns of selection, this is showing you how a population's environmental pressures are showing the population shift. So in a directional selection, right, we have this normal distribution, that's your red line, and individuals that are on the farther side of the curve are showing a higher fitness value. So just like I said earlier, if they have a higher fitness value, they'll probably produce more offspring that look more like them. So you'll get a stack up of traits that have a higher fitness value in that given environment at that given time. And so in this first picture here, this is directional once again. And we have maybe bigger is better in this situation, or hairier, or something along those lines. And the curve is going to shift to the right over time. Now, does that mean it will infinitely keep shifting right? No, not necessarily, right? Because there's give and takes for all things. But that is a directional section. Actually, it could go the other way too. We can see smaller individuals or less hairy and stuff like that. Stabilizing, the second one here, is when you are selecting for the middle, right? We talked about this for birth weight and size of babies. It's usually probably not good to have overly big babies because that might hurt the female giving birth, and she might not be able to make it through birth. If they're too small, they might have developmental issues. So a lot of times there's a median for that species. You know what might happen is we might see a directional shift in a population for a period of time, and then we might start seeing stabilizing, right? Once that population is shifted enough, and it might stay stabilizing for a long period of time. It's all governed by the environment and the population's diversity. And then the last one is what we call disruptive or dispersive. And in this one, the median is really kind of bad, but the two extremes are better. So you might have a black through white and like a grayscale kind of in between, where individuals might have a substrate where they can hang out on black rock and be able to survive, or white rock and they can survive, but there's no gray rocks. And so you end up seeing this disruptive or diversifying selection playing out. That might actually lead to new species fairly quickly, depending on the mode of diversity that's going on in the population. Sexual selection. This is where females choose, oftentimes. Females have a heck of a lot more to invest into a child than a male does. They usually take care of the child, it takes more to make the egg, et cetera, et cetera. So in turn, what we end up getting is what we call dimorphism. Di meaning two, morph meaning shape. So males and females tend to look different. Males usually have to do some sort of display to either obtain a territory or to show off to the female, and then the female kind of chooses. And one of the coolest are the birds of paradise, personal favorite, where they do all kinds of courtship dances, and then the female chooses based on those entities. So it could be just based on pure attraction, or it could be an indicator marker for how fit the male is. If they're not as fit, they might not have as symmetric patterns. They might not look as colorful. They might not be as loud. And the female could kind of select based on those things, right? If they're sick and they have a high parasite load, it might not show as well, and the female can kind of select based on those things. One last couple things is speciation concepts. So there's sympatric speciation, allopatric. So sim means together, patric means family. So within the same family, we start seeing diversity within that population. So a lot of times it's going to be based on genetic abnormalities, where a population is still in the same area, but we have some maybe meiotic errors that allow for diversity within that population. But they're still within proximity that they can actually breed with each other. So just slight alterations in the genetics lead them to not be able to breed with one another. And then allopatric, probably the easier to wrap your mind around. Allo means separate, patric means house. So they're living in separate houses. So this is geographical separation. What I always like to say is like islandness is a big way of thinking of this, right? Where you have I don't even know if that's real word, but separation of locales, right? So whether it's a group that's put on an island physically with land separated by water, or if it's a fish where it's water separated by land, or separation from a mountain range, or separation from a river, but the two populations that have been separated now can't breed with each other for a long period of time. They start diversifying because those two environments, chances are, not the same. And so what's fit in one area might not be fit with another area. And then for some reason, they actually do, a couple individuals from those two locales still come back together in what we call a hybrid zone. They don't breed anymore, right? So defining a different species is the next part and last part of our little lecture series for this one. Oh man, that doesn't show up very well. Hold on a second. See if I can move this, get it to show up a little better. Move it way over here. All right, so we have what we call prezygotic and postzygotic barriers. So prezygotic means before the zygote is formed, these things would prohibit the two species. So I like to call them our thought experiment. You have like blobby number 1 and blobby number 2. They might kind of look different. That's a 2. But we don't know if they're separate species. So we kind of go through this checklist to see if in fact they are separate species. So blobby number 1, you look, oh, hey, it doesn't have as sharp of a tail as blobby number 2. That doesn't matter. Are they living in the same area? If they're living in the same area, they pass geographic isolation situation. Are they living in the same ecology? Meaning, do they live, like if they're in the same area, do they both live in water? Or does one live on the tree? If they pass that one, do they breed during the same time period of the year, temporal, right? Is it one is in the winter and one is in the spring, or one in the AM and one in the PM? That's temporal. Behavioral, do they recognize each other's courtship dances or songs, or even as they talk to each other, communicate to each other? Are they able to actually physically breed with each other? Does the male reproductive organ actually fit the female reproductive organ? That's the mechanical. And then the last piece, if they do pass all of those, right, all, 5, will they actually, the sperm and egg, actually fertilize each other? And if they do, they have passed all the prezygotic isolation situations. If they pass all those, then we still have to run through what we call the postzygotic. And postzygotic, the zygote is formed, but then we have to mark its development. And so the first one is what we call hybrid viability. So in a hybrid viability, we're saying that the offspring of these two blobs, it forms, it starts developing, but its offspring isn't really good. It's not good at maybe camouflaging, or it's not good at producing toxins. And so in turn, its fitness level is really low. So we'd still consider blobby number 1 and blobby number 2 separate. Now, let's just say that the hybrid is okay, right? It actually does have a decent fitness. But then we look at its fertility. Can it actually produce offspring? So this is what we see with like a horse and a donkey producing a mule, right? In that scenario, the mule cannot produce offspring. So we would say that they're sterile. Blobby number 1, blobby number 2 example, they are still separate species. If they still produce a child that passes all these things, the last one was what we call the hybrid breakdown. This is the hardest one to assume. So you go, individual 1, blobby number 1. We cross them with individual number 2, and we produce, let's just call hybrid number 3. Hybrid number 3 is totally okay, got good fitness, produces viable sperm, viable egg. Then you look at 1 versus 2 again, producing another 3. If 3 and 3 can breed together and produce an offspring that is totally still okay, then blobby number 1 and blobby number 2 are in fact the same species. But if that 4 individual fails to thrive, then we have what we call the hybrid breakdown, and blobby number 1 and blobby number 2 are in fact separate species. Now, this is all based on biological species concept, meaning that we're focusing on their ability to survive, their ability to reproduce in a natural environment. That doesn't mean it works for all organisms, because think about bacteria. Bacteria asexually reproduce. You can't use this definitional piece for asexual reproduction. You can't use this for extinct organisms. You're going to have to use different types of things like morphological species concept or other different species concepts. But this is the go-to for trying to understand if, in fact, species have separated from each other or if they are separate species. Okay, and that kind of concludes our evolution and our biotech stuff. The next video will kind of go into some stuff about comparative anatomy. So we'll kind of brush through a lot of the human body systems and connect to the tree of life and looking at the different animal classes.